Hello, this is the family of the phagocytes. Hi, I am the monocyte. Hi, I am the macrophage. Hi, I am the neutrophils. Hi, I am the dendritic cells. Hi, I am eosinophils. Phagocytosis is a specific form of endocytosis where the cell internalizes solid matter including microbial pathogen. It is a mechanism by which microorganisms can be contained, killed, and processed for antigen presenting and represents a vital facet of the innate immune response to pathogens, and it plays an essential role in initiating the adaptive immune response. Receptors on plasma membrane will distinguish particle as a target, and then it will initiate signaling cascade that promotes phagocytosis. There are many types of receptors on a single phagocyte. There are complement receptors, Fc receptors, TLRs, and lectins. The receptors are split into two, which is non-opsonic, which recognize directly molecular groups on surface of target, and opsonic, which recognizes host-derived opsonins that bind to foreign particles and target them for ingestion. Some examples of opsonins are antibodies, mannose-binding lectin, and milk fat globulin.
The first step for phagocytosis process is the activation of phagocytes. Resting phagocyte activated by inflammatory mediators such as bacterial product, LPS, capsules, complement proteins, inflammatory cytokines, and prostaglandins. The circulating phagocytes have surface glycoprotein receptors that increase their ability to adhere to the inner surface of capillary walls. This enables them to squeeze out of capillary and attracted to the infection site. The phagocytes produce endocytic pattern recognition receptors that recognize and bind to PAMPs to attach the microbe to phagocytes. The PAMPs include peptidoglycan, lipopolysaccharide, and many more common microbial molecules. The second step of phagocytosis process is the chemotaxis of phagocytes. Movement of phagocytes to the increasing concentration of some attractants such as bacterial factor, complement proteins, chemokines, fibrin split products released by the injured host cells. TNF-alpha and IFN-gamma drive the formation and maturation of phagosomes. This process triggers phagocytes to produce cytokines, which act as chemoattractants to enhance migration and activation of other immune cells to the site of infection.
Phagocytosis. The process of phagocytosis, attachment of phagocytocytes to the microbes or cells. There are four mechanisms attachment of phagocytocytes to the microbes or cells, which is involve pattern recognition receptors, antibody opsonization, complement protein opsonization, and lastly, neutrophil extracellular traps. The attachment of phagocytocyte can be divided to two, which is unenhanced or enhanced. The unenhanced attachment of phagocytes involve a glycoprotein molecule or pattern recognition receptor called PRR that's found on the surface of phagocytes. They recognize and bind to PAMPs or pathogenic associated molecular patterns. Meanwhile, for the enhanced attachment, which is called opsonization, the IgG sticks antigen of bacterial protein and polysaccharides to phagocytes. FAB portion of IgG bind to antigen with a complementary shape, while FC portion bind to FC receptor on phagocytes. And the other mechanism, which is complement opsonization, when complement pathway activated, defense protein or C3b is produced. C3b bind to one end of bacterial surface protein and other end bind C3b receptor on phagocyte. IgG and C3b is called as opsonin. And the last mechanism which involves neutrophil, where extracellular trapping with NETs. The neutrophil release DNA and antimicrobial granular protein that attach to the DNA, and it will bind to the bacteria, prevent them from spreading, kill them with antimicrobial proteins that attach to the DNA. Next part is ingestion of microbe or cell by phagocyte. Once the phagocyte detects the signal of hydrogen, the polymerization and depolymerization of actin filament and pseudopod out where the cell membrane extends around target, enveloping and pinching off to form a discrete phagocyte to engulf microbes and place it in an endocytic vesicle called phagocyte. During this process, an electron pump brings protons into phagocytes. The pH lowers to 3.5 or 4.0, so the acid hydrolyzes to break down cellular proteins when lysosome fuse with phagosome, the lysosomal hydrolysis. Acidification also releases defenses, catalysin, and bacterial permeability inducing proteins, BPI, peptides, and enzymes that can kill microbes. Meanwhile, for the other mechanism, when the virus and bacteria that invade host cell, they engulf once they enter the cytosol of cells, which is called autophagy. The membrane-bound compartment or autophagosome crawls around the microbes and the surrounding cytosol and subsequently deliver it to the lysosome for destruction. This process also occurs in eukaryotic cells to engulf and degrade unnecessary cellular components.
The last step of phagocytosis process is the destruction of microbe or cell. The phagocytes contains membranous sacs called lysosomes that contains various digestive enzymes, microbicidal chemicals, and toxic oxygen radicals. Lysosome travels along microtubules within phagocytes and fills with phagosomes containing the ingested microbes, and so the microbes are destroyed.
And then all the contents are removed through exocytosis.